Good afternoon, good evening, good morning to you, wherever you might be logging in from around the world. I am Rob Burgess, and I head up business development for Sino Biological. And I would like to officially welcome everyone to the next installment in our series titled Lock and Key Immunodetection Webinars. We have a wonderful and excellent speaker today from Carterra. And he's going to talk about a very exciting technology focused on antibody discovery as well as epitope analysis. Before I get to introducing our speaker today, I have just one very brief housekeeping issue that I wanted to mention. And that is, if you have a question for the speaker, I ask that you type it into the chat box. And then at the end of the lecture, I will verbally walk through as many of those questions as possible within the chat box. So come up with some good questions for the speaker today. Also, please introduce yourselves in the chat box and tell us where you're from. It's always interesting to see where people are logging in and calling from around the world. And so without further ado, it is my pleasure to introduce to everyone today's speaker. It's Dr. Daniel Bedinger. Daniel helped launch Carterra's Lodestar Array platform and now leads the company's global application science team. He has over 2 decades of experience in the generation and characterization of therapeutic monoclonal antibodies, most notably at Soma and Abgenix. Daniel earned his doctorate from UC Davis in cellular and molecular physiology. And it was there that he actually focused on and studied insulin receptor signaling. And the title of Dr. Bedinger's talk today is High-throughput epitope analysis and the latest in antibody discovery from Carterra. So, Dan, it's a pleasure to have you today, and we will now turn over the screen sharing to you.
All right, thank you. Let's get this shared. Okay. Hopefully you all can see these slides now.
Yeah,
so thank you.
Good. Thanks.
All right, thanks for joining me today. So we're going to talk about high throughput characterization of antibodies. So the industry in general has sunk a lot of investment into the optimization of monoclonal antibody generation technologies. And a lot of organizations now have the ability to access large sequence spaces of antibody diversity, whether through phage display, B-cell screening, synthetic library design, AI, and the analytical tools to accurately measure binding affinity and specificity and epitope analysis for a large number of clones really lacks orders of magnitude behind. And so at Carterra, we're focused on trying to develop tools that bring the capability to get accurate analysis at more of a library scale, or at least push the throughput as far as we can on that, because we don't want you to throw a blockbuster candidate away. So typically, binding kinetics and specificity screening has been done on a pretty limited set of antibodies or clones, really due to limitation of conventional label-free biosensors, which make those large analyses either really time-intensive or very sample-intensive. And so typically, people try to compromise by either doing low-resolution assays, with like 1 concentration, 2 concentrations of antigen, limited concentrations, or do significant down selection before they proceed with that characterization. But with automated array-based SPR, we call it HT-SPR, and the LSA platform, you can really get high-quality kinetics and specificity characterization at a new scale, typically at the types of sample levels that people get out of screening campaigns. And this can also be done with supernatants and extracts. So crude samples are often applicable for high-resolution kinetic data in early discovery. Also, the LSA platform can dramatically increase the scale and accessibility of competition-based epitope binning, which we'll talk about quite a bit in this talk. So a little bit of an agenda here. So I'm going to start off talking about how high-throughput SPR can enable and accelerate antibody discovery by giving 3 examples from the literature. I'll do a brief introduction to the Carterra LSA and now LSA-XT platforms, and talk about how we do kinetic analysis with them and then talk about our process for epitope binning and why we think it's useful to do high resolution epitope binning and our assay strategies and analytical approaches, and then going into a little bit more detail, an example of the application of that with the COVID consortium study. So high throughput, high resolution epitope binning is becoming more popular in the industry. The SARS-CoV-2 pandemic coincided really with the broad adoption of the Carterra LSA platform. And the good thing about that was that most of our antibody discovery customers were able to shift when the labs were shut down and things to doing COVID discovery work and still work. And there was a heavy emphasis on publication, rapid publication of SARS-CoV-2 work. So we actually generated quite a body of literature around our technology. I guess the downside of that is that it's primarily SARS-CoV-2 related work, which is great. It's good work and good biology and some amazing demonstrations of the technology, but it's not very broad. And so, we have a lot of customers that are doing a lot of great work in other disease areas, all you would expect, other infectious diseases, cancer, immuno-oncology, neurobiology, all those things. And so I encourage people to check out our symposium presentations at our website, carterrabio.com, if they're interested in some other applications, because we have seminar presentations of that, though not a lot of that work has been published. Okay, so going into the first example here, this is one we love to talk about. The groups at AbCellera and Lilly collaborated very early in the pandemic. They had received some DARPA funding to build a rapid antibody infrastructure earlier. And when the pandemic came about, AbCellera collected blood from one of the earliest recovered COVID patients in Canada and did a B-cell discovery effort where they identified many putative SARS-CoV-2 spike binder antibodies. They expressed many of those, and in the sort of the first tranche, they moved 187 small scale expression clones into Carterra LSA analysis. So both Lilly and AbCellera had LSAs at the time, and they did this work kind of in joint. And so in 7 days, they were able to characterize all of these clones for affinity and kinetics, ACE2 competition, which is neutralization of viral entry, essentially in an in vitro setting, and full symmetrical pairwise competition epitope binning. So they were able to identify the highest affinity neutralizers from a broad epitope diversity. So they could select key clones from individuals or with unique epitope properties. From that, they took 24 into their manufacturability assessment and also downstream potency assays, and then triage that rapidly into manufacturability and production. And they were able to actually file an IND for bamlanivimab 90 days after the start of getting those B cells from the patient. And at 94 days, they were actually in the clinic with that. And bamlanivimab went on to receive emergency use authorization in, I believe it was October of that year, making it the first biologic COVID-19 therapeutic available. So this really shows, I think, kind of the apex. It's definitely the fastest a biologics therapy had ever been developed at this point, and it really shows, what is achievable if companies have the right tools in place. Lilly has a very sophisticated characterization and developability paradigm that they were able to move these things into. And the LSA was a key part about accelerating that initial phase. Another example I'd like to give is a publication from a group at NIAID. This was Josh Tan's group, and they were also making SARS-CoV-2 neutralizing antibodies. And they found they had about 4 discrete communities of neutralizing antibodies against the RBD and 1 discrete community against the NTD domain of that protein that showed potent neutralization. And in some early sort of cocktail studies, they realized that they were only seeing additive potency and not synergistic, but they were more interested in something that would have a real high neutralization with synergy and maybe some different specificity that would give some variant resistance. So they looked into making biparatopic, sort of bispecific DVD-IGs. And that's this format shown here, where you have an IgG construct with two independent binding domains tethered to the end. So by using this understanding of epitope binding, they were able to, in the potency and affinity relationships they understood, they were able to design a discrete set of these biparatopic molecules to take into characterization, knowing that they bound to non-competitive epitopes with each other and should have some bispecific function. When they characterized those, they found some really interesting examples of synergy. The one shown here was the most potent, where you can see in black the potency of the pooled version of the two, or the pooled test of the two clones. And then the biparatopic, or the other two curves, those are replicate data showing more than 100-fold shift in potency with the biparatopic activity. They did some cryo-EM work to try to understand why this was so potent, and they found that there was a really interesting cross-linking created by the RBD. So this set of clones was actually one of the NTD binders and an RBD binder, and it would cross-link across two neighboring spike monomers and lock the SARS-CoV-2 spike protein into an extremely inactive conformation. So this was pretty cool application of that. And the last example I'll talk about here is on actually a different disease. It's still antiviral. Tom Yuan and the group at Twist was looking at ways of validating their platform and demonstrating just how efficient it could be. So Twist has the ability to generate large amounts of synthetic DNA. So they went to the literature and found a paper that had published, a couple hundred, or I think more than 200 sequences of patient-derived Ebola recovered patient antibodies that they believed were Ebola viral protein binders. So in about 14 days, they expressed all of these sequences, cloned them into expression vectors, and started making small-scale, 1 mil transfections of these. In another about 4 days, they purified those antibodies and were able to run them on the LSA for general kinetics of binding, although it's a trimeric antigen, so is avidity, but at least characterized binding, and a binning assay. So they used about a little over 50 analytes, and they included a number of clones that bound to known residues in the literature. And so this enabled them to actually map all of the sequence diversity they pulled out of the publication in just a couple of days to either known binding epitopes or identify them as binding to unique, sort of previously uncharacterized epitopes. So this is a really strong validation of how in a very small amount of time, you can go from sort of unknown sequence space to a fairly high resolution understanding of the binding properties and epitope of these clones. And this could be used to design, cocktails or bispecifics going forward. Okay, so now I'm going to actually introduce the LSA platform itself. The LSA is an array-based SPR biosensor, and it's been pretty widely adopted by the instrument. It launched in the beginning of 2018, and now it's in 19 of the top 20 pharma, many biotechs, numerous CROs, and multiple reagent suppliers. So we actually have two instruments now. We have the original LSA and a sort of updated version called the LSA-XT. I'll talk a little bit about the differences later on. Then we have our software suite, which is 3 software packages. The Navigator software is our instrument control software used to design experiments and collect data. Then we have 2 analytical software packages. One is called Kinetics and one is called Epitope. It's relatively self-explanatory what they focus on. And then we have an entire line of biosensor chips and consumables that you can use to run the assays. So the LSA really excels in many of the core applications of biologics or antibody discovery, those being kinetics and affinity analysis, competition-based epitope binning, peptide or mutant mapping, and quantitation. And so in addition to having a hardware architecture that makes these assays very high throughput, scalable, and sample efficient, we've put a huge amount of effort into developing analytical software packages that make processing these large data files fast and efficient, but also visually rich with lots of tools to access different types of figures and images around the data. The way the platform works is it has two semi-independent fluidic systems within it that address the same chip. So the chip doesn't move, it sits in the center of the instrument. There's a 96 channel flow head where you can flow 96 samples at once, and you can use that to index to 4 locations within the surface to create a 384 spot array. And then the single flow cell will dock over that entire array area and flow one sample over everything. We call this one-on-many analysis. And it's where the really high levels of efficiency for both kinetics and epitope binning, or generally multiplex studies, comes in. So a little bit more resolution on this process. So this is a video showing the 96 needles descending into the sample deck and drawing 96 samples at a time. This print head device docks onto the chip surface and flows the samples back and forth before returning them to the plate. And then it can go pick up additional samples, go to a new location, and print 96 more, eventually creating a 384 spot array, which is actually shown in the picture now on the screen. So it's a high density array. There's some real advantages. We call this continuous flow microspotting technology, and this is kind of our core differentiator. Unlike most microarray systems where you're depositing material on the surface, this is actual flow. So it's going from running buffer to sample, then back to running buffer. So all of the types of workflows that are done on conventional biosensors can be done here, where you're either immobilizing from low concentration samples using electrostatic preconcentration, or you can capture from crude samples if you have an affinity surface. And because we're flowing the sample back and forth, we can maintain a high flow rate for very long contact times. If you have a B cell supernatant with tiny amounts of antibody in it, and you want to capture your antibodies for 45 minutes onto an anti-FC surface, for example, you can do that and allow the surface to build up over time. So we get really high sensitivity that way. And it's quite flexible with chemistries as well. This is just a view of how the array kind of works. The pink vertical rectangles are the 96 channels that are docked. The blue horizontal ones are the 48 interspot references. So those are unprinted locations used for real-time referencing. If you do 4 of these prints from above interlaced, you create a 384 spot array shown below. So the other fluidic module here is the single flow cell. So this docks over the entire area where the array can occupy and flows one sample. So this can be really useful for preparative steps if you're going to regenerate the chip surface or immobilize, say, a capture of a lawn. We call it a lawn when you coat one sample over the entire surface. It is also used to deliver the samples for kinetic injections or competition assays like epitope binning. So that one injection is giving you simultaneous data for all really 432 spots on the array, the 384 active plus the 48 reference. So this is one 270 microliter volume of this injection to get you data in all those parallel spots. Again, it cycles the sample back and forth. So with that relatively small bottle volume, we're able to maintain a high dynamic flow rate to minimize things like transport effects. So again, with the single flow cell, one sample goes over everything, collecting parallel data. Just real briefly, want to talk about our chip types offerings. Much of the chemistries people are used to using on biosensors are available through us. This list is kind of always expanding as we learn and develop new applications, but currently we have a couple different thicknesses of linear polycarboxylate chips, several different carboxymethyl dextrans, including a planar. We have nickel NTA surfaces for capture of histag proteins, several different thicknesses of streptavidin, and protein A and protein AG chips currently available. Okay, with that, we'll start talking about high throughput kinetics and how we approach this. So on the right is a diagram of a very common format for capture kinetics high throughput screening assay on the LSA. So this shows an anti-human IgG surface. This would be made using the single flow cell as what we call a lawn. So this is everywhere. And then you would use the 96 channel side to capture supernatants or even purified antibodies diluted to the surface via their FC. And this could be, any type of antibody fragment or construct that has some sort of affinity linkage, biotinylated peptides or aptamers, Fabs, VHHs with his tags or HA tags. We do all those things. And then this and that uses tiny amounts of sample, way less than 100 nanograms per clone is necessary. Then you inject a titration series of your target with the single flow cell. And the example I'm going to give on the next slide, it's PD1, it's a 17 kilodalton protein, and to generate a titration series starting at 1 micromolar, it uses 7 micrograms of antigen. So very little material for the amount of data you generate. This assay, because this is a regeneratable surface where you can remove the bound ligands, can be scaled all the way up to 1,152 unique ligands in a run. Our instrument has positions for 3 384 well plates on the 96 channel side. And so you can repeatedly load those samples to the array and strip them off. So you can even automate, say, like 3 antigens by 384 well plates. It's only 9 steps in a run. Yeah. Oh, and you can use this to capture high N or dilution series of ligands as well. So you can get high quality data in one go. This is an example of a run that was done on the LSA, set up in an afternoon, run overnight. So 384 interactions. In this case, it's actually only about 38 unique antibodies spotted in 8 to 12 semi-replicates each. That's why you see the repeating patterns. But all of this data, this was the analyte injections you're seeing are 8 wells on a 96 deep well plate. So it used 7 micrograms of antigen to generate all of this. Some cool software features are shown here. So we like people to report accurate results with the LSA. So we do some flagging and annotation of the experimental results as they come out. The ones shown in gray are non-binders that are below a threshold you set in the software. So in your big list of kinetic rates, those would just say NA. If they have a poor fit, a high error, they are flagged, in this case in yellow. And then there's 2 kinetic limitations shown here as the same color, although they could be set to be different colors. If there's a slow off rate that is hitting a limit that you define in the software, it will flag that and report it as a less than function. The affinity is less than 50 picomolar, off rate is less than 2e times 10 to the minus 5, et cetera, based on what your settings are. Also, if you haven't injected a high enough concentration of analyte to necessarily accurately inform the Rmax or the saturation point, it will flag that. Technically it's flagging something that the observed binding level is less than 50% the calculated Rmax, but you can think of it as highlighting to you that if you are going to hang your hat on those affinity and on rate values, that you might want to inject a higher concentration of antigen. It will give you those rate constants, but it flags them for you. So I mentioned earlier, if you don't have 384 things to mobilize in your array, you can increase your N. So this is a really common thing that's done on the LSA, is instead of just getting one value with a goodness of fit parameter, you can actually get mean and standard deviation of equivalent analyses. This is showing one clone in 12 replicates within one experiment. It was actually captured from 3 slightly different dilutions, is why you see the 3 densities. But if you label them with the same name, the software will automatically calculate mean and standard deviation of all the rate constants and parameters from the fit. This could be a really powerful way if you're looking at subtle changes like single amino acid mutations, or maybe a deamidated form, an ion exchange peak of an antibody prep. If you spot it in 12 times at multiple different densities, and you run the same injections of analyte over those 2 preps on the array, you can look, do my error bars overlap, or is there a real difference in the kinetic behavior between those 2 forms? This is a pretty powerful application of the technology. In terms of visualization of this data, the software has some great tools. This is an iso affinity plot where you have the on rate as the y-axis and the off rate as the x-axis, because the affinity or KD value of an interaction is the off rate divided by the on rate. These diagonal lines represent single affinities. So you have 100 picomolar, 1 nanomolar, 10 nanomolar, 100 nanomolar here down these lines. So this is a great way to visualize kinetic distribution of the antibodies in your panel. In this case, some of the clones are highlighted to show all of the replicates that were done. One more example on the LSA of kinetics I want to show was presented at one of our seminars by Denisa Foster from Eli Lilly. They were making COVID-19 antibodies, and this is actually the bamlanivimab Fab form binding to immobilized or captured SARS-CoV-2 RBD mutants. So using a shallow well 96 well plate, they were able to do small scale transfections of many mutants, take those soups, directly capture them to the array, and in an afternoon get the full kinetics of the Fab profile to all of these 96 mutants to the receptor. So I think this is a great example of both crude kinetics and also sort of the richness of data you can get easily on the LSA platform. So I did mention earlier we have a new version of the LSA. We're calling that the LSA-XT. So the same story applies to it as that we like to say about the LSA. You can get 100 times the data 10% of the time using 1% of the sample compared to other leading biosensors. But now we have better than 2 times signal to noise, 2 times better signal uniformity in the array, and 2.5 times faster data collection rate. So what this really translates to is it's the same fluidic system as the original LSA. You can think of it just enhanced optics and some software improvements that make processing the signal data better. The LSA was really well suited, and all of the data you've seen up till now was from the original LSA for protein-protein interactions, antibody screening, affinity characterization. But it was a bit of a struggle for really small analytes or extremely rapid interactions, things that dissociate very quickly. We could get an affinity, but maybe not a rate constant, or things that were very small, were harder to measure. You'd have to get higher surface densities to stay above the noise structure. So the XT sort of brings this to a new level with the faster data collection rate, a little better referencing, and much less noise. So we can do things like kinase inhibitor profiling. So the next couple slides will show some assays where 6 kinases were coupled to an HC30M chip in duplicate, and we injected kinase inhibitors from 3 micromolar and 3-fold dilutions. And this is done in some 3% DMSO buffer. So this is starosporin. This is a 466 Dalton compound. So, for us, very small. And you can see we can clearly elucidate the kinetics to these different kinases. The results match the specificity and the binding rates that we'd expect to see from the literature. another one, sunitinib. This is a 530 Dalton interaction. You can see it has a quite different specificity profile. But again, we're able to characterize both the kinetics and the affinity of these interactions. So we're very comfortable with the LSA-XT saying that analyses down into about the 500 Dalton range are totally doable. And there are some applications where using the array in this one-on-many format makes a lot of sense, like kinase inhibitor profiling. Another application that we've been working with on the LSA-XT is FC receptor binding. Some of these, they're not small proteins, so it's not a signal limitation, but they do have a rapid dissociation. So the faster data collection rate of the LSA-XT makes this nice. It's also very high throughput. You can put down many FC receptors into the array and inject your antibodies of interest and collect all that data in parallel. So this greatly speeds and significantly reduces the materials required for these assays based on the experiments our customers were doing in the past. Also, I will mention Sino Biologicals has a quite robust, I think about 30 FC receptors, many of which are biotinylated in their catalog. So that's probably a great source to build an assay like this. So this is just some data. These are 11 human and Sino FC receptors spotted in 8 replicates each. So each row is the replicates and each column contains the different receptors as you go down. So you can see we get really good agreement across the replicates. We're able to accurately measure both rapid and high affinity, so low affinity and high affinity receptors. And this is everything you're looking at here, if you remember, is 1 injection per concentration of the antibody. This is trastuzumab. So very little analyte is required for these experiments to generate such robust data. If we zoom in on the data, you can see we're able to get really nice fits for both subnanomolar and micromolar interactions. Okay, and with that, I'm going to switch to talk about epitope binning. So why do people want to do epitope binning? The function and the mechanism of action of therapeutic antibodies is linked to its epitope. The affinity of an interaction can be pretty readily assessed and engineered or optimized, but the binding epitope is innate, and you can't take an antibody typically that binds to one epitope and mutate it to bind to a different one. That's really kind of the core part of what that antibody is. And so early epitope characterization can serve as a surrogate for functional diversity. Sequence diversity is one thing, but you can have a fair amount of sequence space that reflects a limited epitope space. So it's good to verify that in addition to sequence diversity, there is epitope diversity especially in a new discovery panel where there's interest in multiple MOAs or maybe combinations, bispecifics, cocktails, things like that. You can also use high throughput epitope binning data to inform large sequence sets. Say you did a campaign where you ended up with 3,000 putative binding sequences. You can probably use bioinformatics to organize those into family trees where clone sequences are relatively related. So say you took 300 relatively unique sequences and ran a full-scale epitope mapping or epitope binning experiment, you could then bucket a large portion of that sequence diversity into expected epitope space. It can also be used to identify sandwiching pairs or establish IP if there's publications or patents around certain binding profiles and epitopes. The more clones you include, the more likely you are to show a difference. So the assay setup for epitope binning on the LSA scales linearly. So if you add one more clone to the assay, it's one more well on your ligand plate and one more injection of analyte and sandwiching antibody in the final assay. Most other platforms, if you try to work out the logistics of scaling these pairwise competition assays, they scale sort of exponentially in complexity and typically are limited around 20 to 30 clones maximum. Where on the LSA, we can scale all the way up to potentially 384 by 384 in one automated run. I'd probably recommend breaking it up into a couple where you have a 384 spot array and you only do 180 ligand or analyte injections for each one and then merge those files together. But those are kind of details. But this generates a huge amount of data. If you were to do a 96 by 96 competition binning assay, that's over 9,000 interactions. If you did 384 by 384, that's almost 150,000 interactions that would show up in your heat map. So each unique interaction can be thought of as a probe. You can imagine if you have 4 antibodies and you bin them together, you can tell whether they compete with each other, but you're not going to have very good resolution about whether there's any subtle differences in that. But if you have a diverse set, so you have 96 antibodies that bind to a wide range of epitopes, you start to uncover subtle differences between them based on their shared competition profiles, because each antibody that you inject or use in the assay can potentially reveal differences in the others. So the LSA epitope binning workflow is kind of outlined here. The process starts by immobilizing the clones onto the array. And then we have 2 assay formats that we typically use. The one on the top shown here is what we call the premix format, where you inject either the antigen alone or the antigen in the presence of a potential competitor antibody. And if it can sandwich, you see additional binding. If not, it will inhibit the binding. This is really useful for multivalent analysis. If you have a dimer or a trimer antigen, this is the preferred format. If you have a monomer, you can use the one shown on the bottom, which is called the classical binning assay. And this is where you inject the antigen and then subsequently inject the sandwiching antibody and look for a gain of signal if you have a sandwiching interaction. So once those experiments are run, we have analytical software where you define cutoffs and thresholds, and then the software will automatically make and sort these heat map and these network plots that show how the epitopes relate to each other of these clones. A little bit more on the binding cycle here. So this is a classical binning cycle where you have a baseline followed by an antigen injection. Then there's the green bar is actually a normalization bar where it scales the binding of that antigen to 1. So if you were to have, say, some loss of surface activity over time or some variance there, it still allows you to set global cutoffs because it does a normalization. The second bar is a report point bar, and it's looking at sort of the average value of the signal under that bar, and that is what is used to populate the heat map, and it's actually the difference from the blue control injections. So even if you have dissociating antigen, you can still set cutoffs for that. And then it allows you to position cutoffs, and everything that crosses the report point bar in the green is a sandwicher. Everything that crosses it in the orange is a blocker. And these can be adjusted on a per ligand basis if needed. So the analysis software interface is shown here for the epitope binning. And this is really powerful because you have 3 views that are all interrelated. So there's a sensorgram view, there's the heat map view. So immobilized antibodies are rows, injected antibodies are columns. If they're sandwiching, they're green. If they're blocking, they're red. And if they're self versus self, they have the dark outline. And then we have the network plot. In this plot, every clone is a node, a circle, or a square. If they're competitive with each other, there's a cord connecting them. And if they have the same competition profile, they're clustered into one of these colored epitope bin regions. And these 3 plots are interactive. So in this case, this cord was clicked. It highlights the cells in the heat map related to that relationship and shows you the sensorgram. So this was an asymmetrical interaction. So this allows you to dig deeply into your data, explore the relationships in the heat map and the network plot, and see whether you believe all these calls or if you need to adjust your settings to properly interpret the data. This is really an enabling feature to work with these large data sets. So I mentioned the bin network plot, where if the clones have the same competition profile, they are clustered into one of these groups. Another thing the software does automatically when you sort these heat maps is it generates a dendrogram. So this shows the level of shared competition profiles across clones as you go up. There's an option to set a custom community threshold where you can define basically an arbitrary cutoff height and define more generalized bin communities. And this is really useful. We'll talk about this a lot in the COVID example at the end. Also, there's some great visualization tools where you can color your data by orthogonal data. In this case, that's showing whether or not the clones are mouse cross-reactive, what library they come from, or what subdomain they bind. You can also do this numerically with potencies and affinities. And this is just sort of a toggle in the option after you load in tables of data. One more thing on epitope binning, a common application is to look for sandwiching pairs. If you're doing, biomarker analysis, PK/PD studies, or looking for infectious diseases, you need to find highly efficient sandwiching pairs. So on the LSA, typically in 24 to 40 hours, you can take a panel of clones, create an array, do a kinetic analysis to the target to figure out on and off rates, and then do a symmetrical pairwise competition binning, where you'll see, which clones sandwich well together and which ones don't, and even identify which ones are likely to be more efficient capture or detections based on their behavior. And this typically translates really well to other sort of plate-based or more conventional immunoassay formats. Okay, with that, I'm going to move on to talking about the COVID study. So this was a large collaboration study run by La Jolla Institute for Immunology, and I think it's one of the richest examples of epitope binning data being overlaid with structural data. And I was a part of this, and it was published in Science. So the COVID consortium was a global nonprofit consortium funded by the COVID-19 Therapeutic Accelerator, Bill and Melinda Gates, and some others. As I said, run by LJI, and it involved a number of labs around the world collaborating to characterize antibodies that were contributed to the consortium that neutralized SARS-CoV-2. A number of biologies like viral neutralization, FC receptor interactions, viral escape were all looked at, but two of the groups, Duke and us at Carterra, were using the LSA to do affinity and epitope binning characterization. So about 75% of the 400 antibodies that were contributed to this study bound to the SARS-CoV-2 RBD, which is the receptor binding domain. And there was a small set beyond that that bound to either the N-terminal domain or only full intact spike. So the binning I did, I'm going to talk about here, is for the RBD binding clones. So these clones all bound to a shared globular protein that we could express, or that LJI expressed as a separate entity. As I mentioned, there was an affinity component to this study. The group at Duke was primarily responsible for reporting this data. You can see they generated affinities to the RBD, which was a monomer and gives discrete one-to-one kinetics and quite a bit of kinetic diversity across the panel from the COVID. And then they also looked at spike binding, and this is a trimer with some avidity. So the kinetics are slightly less descriptive, but still illustrative of the binding properties. So I did the epitope binning of the entire panel. This is a representative assay from that. This is 170 ligands by 188 analytes, or almost 32,000 interactions shown. This is a single experiment, and this is really the way you would like epitope binning run at this scale to look. The matrix is highly symmetrical, where you're getting equivalent competition generally in both orientations. And we're able to find some really discrete shared behaviors to make some nice communities. So I'll highlight a couple of those here. So this is a community that was identified. You can see that they're competitive with each other, and they share many competition and sandwiching relationships, and it's generally coherent how they do it. This purple community is a closely related community, but it's different in that it has clones from these other, the yellow and blue community are sandwiching, where in the yellow community, they were blocking. So even though these clones compete with those and they share many relationships, due to the resolution of this assay, we're able to say this is a shifted epitope with a different behavior. And you can see you can keep going on highlighting communities that have additional properties and differences. So this is how this is applied. We try to find coherent sort of breaks in the data where there's obvious shared behavior. So this was done, this is the dendrogram and some community assignments from the COVID consortium. LJI did cryo-EM on selected antibodies from these various communities. Over 40 antibodies were evaluated for structure, and they found that the binding interface from cryo-EM was highly related to the competition epitope cluster bin. And so we sort of mapped it here on the RBD. You can see where the different communities been. The dotted line is the ACE2 binding site. So you can see we sort of move around various faces of that as these epitopes shift. So this was really interesting, really great confirmation that these communities, if applied strategically, can really translate direct to discrete structural domains. It was interesting that there was some patterns with variant resistance from the different communities. You can see the ones that are dark had high levels of neutralization. And all the way through delta and mu, there were clones that seemed to have real pockets in certain communities of resistance and branches on the dendrogram. As the Omicron strain developed and the number of mutations increased, this became hard to maintain for many of these antibodies. You can see that if we include the BA 1.1 and later Omicron strains in this, that it becomes a bit more stochastic, which ones are resistant. There's still some little pockets of resistance, but this is really high sequence diversity to expect these antibodies to bind. So out of the entire panel, there were 66 neutralizers that maintain Omicron resistance. LJI published a second paper looking at sort of the mechanism of this resistance, and they found some interesting things in that, I think I'll talk about in the next slide, that the clones that were antibody derived and bound to the RBD, all of the ones that had potency did so with avidity. They would bind to two monomers of spike within a trimer, much like the biparatopic antibody that was shown earlier from NIAID. There was one subdomain, RBD4B, which actually still maintained potent neutralizers with monovalent binding. And it's interesting that in that region where those clones bind, there's 3 mutations in some of the Omicron strains. 2 are conserved and 1 is right on the edge. So those clones are able to sort of bind around the mutational set and maintain potency, whereas the high avidity in the other ones seems to maintain some potency, as you know, the affinity is kind of enhanced, and I think there's structural limitations to how the spike can move when bound bivalently by an antibody. So this is another real interesting paper that came from this work. And with that, I'm going to thank the contributors for these, you know, the team at Carterra, the folks in the COVID consortium, especially Sharon Schendel, Catherine Hasty, Hua Yang, and all the COVID participants. And I'll end with one final summary slide. You know, I think high throughput epitope binning is broadly becoming an integral part of early antibody discovery projects. Epitope competition data is crucial for selecting antibodies and for cocktails and bispecifics. The LSA really allows you to run kinetics at a scale that is unrivaled. And then I want to thank Sino Biologicals for the invitation to present today.
Thank you, Dan. That was an excellent presentation. We really appreciate it. Wonderful technology, lots of great data. I'll go ahead and jump right into the questions, as it looks like we're getting a little bit short on time here. The first question is from Rahela Toysakani. And Rahela asks, is it possible to use cell lysate as a source of antigen for epitope binning?
It's possible, yeah. As long as there aren't other components in the media that are binding heavily to the surface or the antibodies. I've actually done quite a bit of work like with kinetics, with solubilized cells, to look at membrane protein and binding thing. If you have a mixed micelle detergent lipid, you can have a high expressing cell line, lyse it up, spin out the particulates, and then inject that as a titration series of antigen. And the antibodies should have sufficient specificity to recognize that component. So that can definitely work.
Great, great. And another question from Rahela is, can you use phage antibodies instead of actual purified antibodies for the epitope binding studies?
So phage display derived like SCFEs or VHHs in the way most people make periplasmic extracts or bacterial supernatants tend not to be high enough concentration to use in epitope binning. You can absolutely get kinetics with them because if they have an expression tag, you can capture them to the array and inject antigen. But in epitope binning, there's a component of injecting the analyte over the surface where you really need sort of a molar concentration of that analyte high enough to drive binding. If your expression's good and the clones bind with very high affinity, then you might be able to get away with it. But typically, if you have a diversity of affinities and a diversity of expression levels, those assays would be really hard to interpret because you might see very small binding signals in that sandwiching response. It's just due to the concentration of analyte in solution isn't high enough to drive that binding or block the binding sites in a premix assay. So typically, you're better off using either purified or high expressing samples for those. Things like XP293 transfection soups have plenty of antibody in them to use in epitope binning, or even hybridoma soups, but bacterial extracts tends to be dicey.
Great, thanks for that answer. Yushin Hao is asking, can you perform kinetic analysis for epitope competition data?
Not sure I mean, those are kind of separate applications. There's, you can look at binding kinetics to the epitopes you're interested in. The competition studies are typically not used to interpret kinetics. Usually that's part of the same workflow, but a different set of experiments. We do have some applications around PROTACs, the targeted protein degraders, which are heterobifunctional molecules that are binding to two components. And we measure affinity of ternary complex formation versus the two individual binary complexes. And you can calculate things like cooperativity or inhibition from those by comparing the rate constants. So maybe that's what you're thinking.
Yeah, I think that probably answers the question there. And Wen Guo is asking, how is a neutralizing antibody recognized by LSA?
Yeah, so typically when people are doing neutralization experiments on the LSA, it's that if you're looking for an antibody that will inhibit, like say, a receptor ligand interaction, if you bind to a receptor, can the ligand of that receptor still bind? Or in SARS-CoV-2, it was if you bind to that RBD or that spike molecule, can the ACE2 target still be active? So it's a pseudo neutralization study, but people do it quite a bit, and it tends to translate relatively well to cell-based conformation assays if you're looking at those simple sort of blocking type mechanisms. Obviously, there's other mechanisms of neutralization that would be much harder to tease out with this type of application, like if you're dimerizing a receptor that causes it to be inactive on a cell, you might not see that if it still allows ligand binding.
Great. Thanks for that. My colleague Amy Sheng at Sino is asking, is there any buffer restriction? For example, is glycerol or trehalose a problem on the platform?
Yeah, so the system is chemically compatible with those solutions. You always, when you're running SPR, you need to be cognizant of refractive index mismatches between the sample and the analytes that you're injecting. Because if you get very large differences, we call them bulk refractive index shifts, it can make it hard to interpret the data. But if you had samples that you couldn't remove the trehalose in, what you would want to do is have a running buffer that matches that refractive index. And you can do simple tests by doing a buffer injection to see if you get a big response or not. So you can add, trehalose to your buffer or match them. Like we do a lot of experiments with small amounts of glycerol or DMSO or other things that do contribute refractive index, but we want to match our analytical samples to that, especially for kinetics. It gives a much cleaner result if you can match those.
Right. This question is from Raghu Sangana Gary, and it's about really the universality of your epitope binning software. Can that software be used with other instruments, or it's only restricted to the Carterra instrument?
So the software is developed, to mesh directly with the LSA data, and it's only available to customers that have an LSA. That being said, there is some capability to import, like Excel-based tables of data in there and do some processing and make network plots and stuff that sometimes our customers take advantage of. If you're interested in the technology in general, one thing I didn't say earlier, if you want to see LSA data or try it out or learn more about the platform, reach out to us. You can contact us at our website and things.
Jazia Shafiq is asking, how many times could a chip actually be used? So the chip has the same ligand on it, like with different antibodies. Can you do like multiple screens, I guess?
Yeah, so the capture surfaces, like if you make an anti-human FC or use a protein AG or a nickel chip, they can be used quite a few times. You can strip them and reload them, even take them out of the instrument, store them and reload them a few times, not indefinitely. Assays where you covalently immobilize an array of ligands, like you would do for an epitope binning experiment, those tend to be more single use. You use them. You can use them extensively while they're in the instrument, but once you sort of pull them out and make other arrays, they're kind of done. So,
right. I have a question here. I think everybody would be interested in. I'm just wondering if you could talk about the advantages of using SPR technology as opposed to plate-based technology like ELISA. I mean, why is it so much better than an ELISA system?
Sure. I mean, for things like early kinetic characterization, you're really learning a lot about the system, rate constants, affinities, and it's easy to include things like specificity screening. If you have multiple isoforms of a receptor or off-target proteins, those can all be incorporated into these initial screen runs. And so rather than sort of an iterative process where you find hits, select them, scale them up, and then come back to it to try, okay, now let's understand the affinity and select leads. You really have all that information right at the beginning. So you can jump right ahead to sort of putative lead selection or, move smaller amounts of clones into downstream assays than you might otherwise be able to do. Also, things like epitope binning, it scales really easily on the LSA. 96 by 96 binning is 2 plates, a plate of ligands, plate of analytes. On an ELISA, it would be like 100 plates.
Right. Just much more high throughput and capable in that respect.
Yeah.
Thank you, Dan. I appreciate that. We've run out of time. I apologize to Chunman Chen and many others who I didn't get to your questions today. Please email those over to Sino and we'll forward them on to Dan and others at Carterra, get answers for you. Let me just go ahead and briefly thank all the attendees. We had a huge turnout today. Appreciate everybody attending. I just have to say this because it's very interesting where everybody attended from very quickly. We had folks from Egypt, Germany, of course, all across the United States, the UK, Poland, Canada, India, Portugal, Yemen, Switzerland, Iran, Nigeria, and Ukraine. So thanks for a wonderful attendance. Certainly, Dr. Bedinger, we appreciate you lecturing today under the Sino Lock and Key webinar series That's fantastic work. Congrats to you. And congrats to the entire Carterra team on the exciting technology and the wonderful success that you've achieved.
Well, thank you very much. Thanks for listening, everybody.
Yeah, and one other person, my colleague Max Bleckman, who organized and executed this talk today. Thank you for all your hard work and your efforts. It was another successful webinar. And with that, I will say good night, good morning, good evening, wherever you are. And that completes this week's webinar. Thank you.